Good morning, good afternoon, and a good evening to everyone present here today. I would welcome you all to our Life Sciences webinar series, this being the first webinar out of many scheduled for this year. My name is Sahil Gulati. I'm the Applications Manager of Life Sciences at Emitech Gatan. Before I highlight a few things and introduce our speaker for today, I will briefly go through the question submission process. You can type your questions in the question panel of the GoToWebinar task window. We will really try to answer all your questions immediately, depending on the time remaining after the talk. But if by any chance we run out of time, then we will take the questions offline, or we'll be answering to your questions via email. So starting with a few words about Gatan and what we really do. Now, as you already know, Gatan is the world's leading manufacturer of instrumentation and software that is used to enhance and extend the operation of electron microscopes. Speaking about the product compatibility, Gatan products are fully compatible with nearly all the microscopes present in the world today. We covered an entire range of research processes, starting from specimen preparation to data collection to data analyses. The Gatan brand name is recognized for developing and delivering high-quality products and leading technology for more than 50 years. Now, apart from specimen preparation, Gatan focuses on two major research areas. The first being material science, and the second being life sciences. Focusing on life science research in today's webinar, we have a greater portfolio of life sciences products, and these products include cameras such as the K3, which you already know is a successor to the famous K2 Summit camera. K3 has the largest field of view of 5760 by 4092 pixels and offers a frame rate of 1500 frames per second. And therefore, K3 provides the maximum throughput that you can obtain from a camera, giving you more than 1,000 very high quality stacks or movies in a given hour. BioContinuum is our latest energy filter that comes with a K3 camera and provides both extremely stable optics and very stable VLP positioning with respect to time. Now, since BioContinuum comes with a K3, which is basically held after the filter, it provides the highest imaging throughput as well, same as what K3 will provide you, but after energy filtration. K3 base is a variation of K3 camera with a smaller sensor size of 3456 by 4096 pixels, providing lower throughput when it compares to K3 camera, but several-fold higher throughput as compared to our direct market competition. Now, touching on the next generation cryo holders for high-resolution cryo-electron microscopy and tomography experiments, ELISA holder provides extended hold time for liquid nitrogen, enabling the community or users like yourself to collect uninterrupted data for more than 9 hours without having to fill liquid nitrogen in the holder. Additionally, it offers centrosymmetric design, which reduces liquid nitrogen settling and drift whenever the stage is moved or tilted, because it maintains the center of gravity because of its centrosymmetric shape. And overall, everything results in high-resolution structures, even on lower-end microscopes, which has a side entry holder. Now, this was about the hardware side of Gatan in life sciences. So let's talk a little bit about the software solutions for automated data collection. Now, Latitude S is for single particle data collection, and Latitude T is for tomography data acquisition. One of the most important features Latitude now offers is image shift beam pill compensation scheme, which simply increases the image throughput to more than 4-fold. So what you're doing in image shift beam pill compensation scheme is that you're minimizing the use of stage movements and maximizing the use of optical movements of the microscope, again, improving your throughput. Secondly, Latitude provides Python support along with the scripting. which provides users an opportunity to modify their single particle or tomography data acquisition workflow. Because as you know, some samples are more complicated than others. So therefore, they might require special treatment. You might want to check the thickness of ice. In all those cases, Python scripting comes really handy. Thirdly, Latitude S, along with Serial AM, because they both are very optimal with K3 and biocontinuum, provides the highest throughput from, again, K3 and filtered K3 versions, making these softwares extremely powerful in increasing the productivity of the cryo facility that you're part of. And finally, and one of the most realized and user-like features that Latitude offers is its extremely user-friendly workflow that essentially results in zero downtime and immediate productivity. So just to give you an example on how easy it is to mark hold positions for data collection in ImageShip bean-till composition scheme of Latitude. So basically what you do is you click auto-find. You enter maximum ImageShip that you'll be needing depending on the microscope. You enter the hole size depending on the grid you're using, and you click find. And it centers automatically and marks all those holes for data collection. Then you move to another position, and then you click find again. Again, it centers the holes and marks all those positions for you automatically for data collection. And again, you do exactly the same thing all over the grid process, all over the whole grid itself. And that means you can do things automatically today and reduce the manual intervention. Most importantly, Latitude avoids thick ice areas or ice contamination, as shown here automatically. Again, maximizing automation and minimizing manual intervention so that you can schedule your tasks within 5 minutes, and by the time you come tomorrow, after the overnight data collection, you have thousands of images waiting for you to process data on. Now, with all that brief introduction, I would like to introduce our speaker for today. I welcome Lexi Walls, who is a scientist at the University of Washington. Lexi is one of the heroes, I'm very proud to say that, researching on SARS-CoV-2 virus to combat COVID-19 disease. Her work has been foundational for understanding coronavirus structure, especially the spike protein of coronavirus, or otherwise called as the G protein, to understand its function and developing therapeutics before and during the pandemic. She has co-authored more than 28 publications. She has contributed to 4 US patents, and her work has led to the development of 2 clinical trials, and she'll be covering that data in her presentation today. Just a little bit of background on Lexi. Lexi is from Massachusetts, and she received her bachelor's in biochemistry from the University of Massachusetts. She moved to Seattle to pursue her PhD with David Wiesler at the University of Washington. And whenever she is not working on SARS-CoV-2 virus or any other coronaviruses, she spends her free time hiking and climbing mountains. So she's pretty active as well. She enjoys nature, which is awesome. So again, before I give the electronic podium to Lexi, please remember to submit your questions by typing into the question panel of the GoToWebinar task window. And with that, I will leave the electronic podium to Lexi. So please, Lexi, go ahead.
Hi, everyone, and thank you so much for that introduction and for having me today. And I look forward to discussing after my presentation. If anyone has any questions, I'm happy to answer them. Okay, so like Sahil said, today I'm going to talk about coronaviruses and specifically SARS-CoV-2. And what I'm really going to focus on is the work that myself and many of my coworkers in the Wiesler lab have been working on to really try to visualize what the coronavirus spike looks like just on its own, and then also in complex with neutralizing antibodies, and really how we've taken that work and pushed this to the clinic. But before I get into that, I just want to back up for a second and talk really about kind of the background of coronaviruses and where we were at the end of 2019 when the pandemic was really beginning. And so a study was released essentially in October of 2019 saying that, no country, including the US, was fully prepared for a pandemic. And this report went largely, untalked about, undiscussed. And unfortunately, we're all still currently living the consequences of these types of things. And at the end of 2019, there were reports stating that there was kind of a novel virus that was kind of being discovered and people were trying to understand at the beginning of December. And then towards the end of December, scientists in China were really starting to figure out what was the cause of this novel pneumonia, that at the time, there were low numbers of people infected, and really we had no idea. This was completely novel. And the first thing that they did was to rule out SARS coronavirus. So SARS coronavirus was a infection that jumped from animals to humans back in 2002. This was really quickly brought under control. There were no more SARS infections after 2004. SARS was not the first time that coronaviruses jumped from animals to humans. There was actually another instance of this in 2012 with MERS, or Middle East Respiratory Syndrome. MERS is still an active infection with a few hundreds of cases per year. And until late last year, there were no therapeutics, no vaccines, nothing really to combat human infecting coronaviruses. And I'll talk to you all today kind of about the work, and a lot of effort has happened globally to really change the interface of how coronaviruses are therapeutically handled. But going back to the end of 2019 and kind of the beginning of 2020, when scientists were trying to figure out what type of virus was this. And as soon as they looked at it and started to sequence it, they realized that this was a coronavirus. It was not SARS, or severe acute respiratory syndrome, the original kind of example of a crossover from animals to humans that ended up being really deadly in the modern world. But what they identified was a novel severe acute respiratory syndrome coronavirus 2. So now we call it SARS-2. And coronaviruses are really beautiful when you look under the electron microscope. And they're actually named corona for this crown of spike glycoproteins that you can see protruding from the edge of the virus. That's really where they get their name. And what I've been working on for the last 6 years is really to focus on this spike glycoprotein. And there's a number of reasons for doing this. One, that when we started looking at this, there was no structural information about the spike glycoprotein. And the reason that we really wanted to find structural information about this protein is this is one of the only proteins on the surface of coronaviruses that the immune system really responds to. So majority of neutralizing antibodies are really going to this protein. And so we wanted to structurally characterize it. We wanted to understand, how do neutralizing antibodies interact with this protein? And what does that look like at the molecular level? So we've been using cryo-EM with a mixture of crystallography to really understand what are those interactions and how can we dismantle this spike glycoprotein. And so that kind of brings us to where we are today. And so we all have been living with this global pandemic of SARS coronavirus 2. And this is really an old graphic. It doesn't matter how often I update this, the cases are never correct. But just to show you a sense of really how global this virus has spread, and how we're all still kind of reeling with those effects. Now, before I talk about any of the work we've done on SARS coronavirus 2, I just want to show you kind of a summary video that was made by Janet Owasa, who's a really talented scientific animator. And this video is really showing kind of what we knew at the middle to end of 2019 about structure of coronavirus spike proteins in general. And so what I'm showing here is the spike protein. It's a trimer and it's heavily glycosylated, and you can see that it's present on the surface of the virus. Now, what's really interesting is that these receptor binding domains, which you'll see in a second, are actually mobile on the surface. And so they're going between this up and this down conformation. And they're only able to bind their receptor when they are in this up conformation. After binding the receptor and undergoing a proteolytic activation event, which you could see in orange, the spike protein is able to go from its pre-fusion conformation, or how it exists on the viral surface, into its post-fusion conformation, which you can see depicted here. Now, the purpose of this rearrangement, this amazing structural rearrangement that the spike protein undergoes, is to bring together the viral membrane, which you can see on the bottom, and the host membrane. And this bringing together of these two membranes is really what allows the dissemination of the virus genetic material into the host cell. Now, a lot of work has been done to kind of identify and understand what that means for, the SARS coronavirus 2 spike. So this video was based on a number of other structures prior to 2019, like the structure of SARS coronavirus spike, of MERS coronavirus spike, and of previous structures solved like MHV or HKU1 as well. And so kind of at the beginning of 2020, as soon as the sequence was released and everyone was able to start characterizing and identifying the SARS coronavirus 2 spike protein, we really got to work to really characterize the structure of the spike. And before I talk about how we characterize the structure of the spike, I want to point out something that we all are hearing a lot about with regards to the current vaccines that are out right now. And so this paper came out in 2017, so long before the pandemic. And this was done by a number of groups, including Andrew Ward, Jason McClellan, and Barney Graham's group. And what they identified was really this segment of the spike protein. And so this is really in the interior of the spike protein. And they identified a segment with a flexible loop between two helices. And now what's important about this loop is that in the pre-fusion conformation, as it's shown up here in figure B, this loop is connecting two separate helices. But when the spike refolds into the post-fusion conformation, these helices combine into one long helix. And so what they postulated was, if you could put two prolines right here in that flexible loop, you could potentially prevent the refolding of the spike protein into this long, like one of the pieces into this long helix. And that's exactly what they've shown. And that's exactly what everyone in the coronavirus structural biology field has really adopted ever since this paper. And it's even what many of the vaccines that many of you may have had the opportunity to receive also contain in their sequence. And so what you can see, what they show is that, when you express just SARS spike wild type, you get a mixture of this post-fusion, this really long elongated rod, and of course the pre-fusion spike protein, which is this triangular globular trimer. However, when they express with what they're calling 2P or 2-proline, what you can see is that nearly 100% of the protein is in this pre-fusion conformation, so as it exists on the viral membrane. And they've also done a lot of characterization showing, it's much more stable, so you can get higher yields, and this is great for a number of reasons. So we took this knowledge and we applied it to SARS coronavirus 2, as many others did as well. And I worked really closely with a staff scientist in the lab. His name is Young, and we together solved the structure of the SARS coronavirus 2 spike protein. And so what I'm showing you here is just a raw micrograph and 2D class averages of the spike protein that we first solved. And at this point, we were working really, really quickly to try and get information as quickly as possible. So literally the exact same day that I purified this protein, I handed it off to Young and he froze instantly. And so what you're looking at is a continuous thin carbon grid over lacy carbon that he collected on the Krios with a K2. And what he was able to get from this beautiful data set, we collected quite a large data set. We had a little over 4,000 images. And what he was able to see is truly the structure of the SARS coronavirus spike protein. And you can see that it's a trimer, so each monomer is colored in a different color, in blue, yellow, and pink. And what we found from this data set was that we had a large number of the spikes in a closed conformation. So all of the receptor binding domains are in a down conformation. In the exact same data set, we were able to pull out some up receptor binding domains, which I'm highlighting here with an arrow. You can see that this blue domain, this receptor binding domain, is slightly ajar compared to the version on the left. And this was a little different from what we had seen previously with SARS coronavirus spike protein and MERS coronavirus spike protein. Those two spikes we really saw in majority of open conformations. So mostly we had 2 receptor binding domains up. We even had 3 receptor binding domains up. But SARS-2 definitely the spike protein seemed a little more closed compared to those. Other than that major difference, though, we really wanted to look, from at the protein level. SARS-2 is very closely related to SARS coronavirus, the original SARS coronavirus. It's much more closely related to other, bat or animal coronaviruses, but from what we had structurally at the time, SARS coronavirus was really the closest spike that it was related to. And largely, other than that difference in the receptor binding domains, in the up versus down propensity of the receptor binding domain, the organization of the spike protein and largely all of the domains are really similar between SARS coronavirus 2 and SARS coronavirus 1. The organization of the receptor binding domain subunit, so the S1 subunit, we still have this nice V shape, and the S2 subunit, which is even more highly conserved than the S1 subunit, is structurally virtually unchanged. Now, at the beginning of 2020, even though we knew that SARS coronavirus 2 was most closely related to SARS coronavirus 1, many people were postulating, what the receptor binding domain, what the receptor, was of this novel coronavirus, and it was later shown structurally and also biochemically that the receptor is ACE2. Now, this receptor is exactly the same as the receptor that is used by SARS coronavirus 1. And interestingly, it's also the exact same receptor used by another human infecting coronavirus called NL63. NL63 causes common cold-like symptoms in people, and it's virtually impossible to find someone who has never encountered NL63. So we've all previously been infected and in general just had common cold-like symptoms. So SARS coronavirus 2, we know, uses the ACE2 as its receptor. And one thing we wanted to kind of identify as we're thinking, we have this pandemic underway, and we're really trying to identify, okay, where are the neutralizing antibodies that target this SARS coronavirus 2 spike going? Where can we find potential solutions to these infections? And so we teamed up with Humabs and Vir Bio, a company that's split between the US and Italy, and Switzerland. And we really started to identify where antibodies from people who had previously been infected were targeted. And so that's what I'm showing on this next slide. So all the way on the left, we have antibodies from hospitalized people, from symptomatic people, from asymptomatic but known positive cases, and from samples previously taken before the pandemic. So they should never have encountered SARS coronavirus 2. They may have encountered they probably encountered other coronaviruses, but never SARS coronavirus 2. And what we can see is that there are really a lot of, so this is an ELISA. What we can see is there's really a lot of antibodies going to the spike, with more antibodies in a hospitalized cohort than in the symptomatic, and then even less so in asymptomatic. And what we can further see is, as we kind of narrow into the spike, that the majority of the antibodies that are targeting the spike are likely targeting that receptor binding domain, so the portion of the spike that really interacts with ACE2. And what we can further show is not just that majority of the antibodies that are targeting the spike bind to the receptor binding domain, but also that majority of the neutralizing potential of these antibodies is held in those that bind to the receptor binding domain. So what we can do is we can show that if we deplete all antibodies or close to all antibodies that bind to the receptor binding domain, we can show, previously we have a large number of antibodies binding to the receptor binding domain. Following depletion, we've lost, 100 or 1,000 fold of those antibodies that bind. And then we can look at the exact same samples in neutralization. And so you can see that previously these samples were able to potently neutralize the SARS coronavirus 2 pseudovirus. And then following depletion, we're losing about 90% of the ability to neutralize the virus. And this has been shown by many other groups since this paper came out. And what this means is that majority of the ability to neutralize SARS coronavirus 2 is coming from antibodies that are targeting the receptor binding domain. And so this was really exciting. We started to have an answer of where we could target to really understand the immune response. However, when you look at sarbecovirus spike proteins and you look at the structure, unfortunately, the most conserved region is down here in what we call the S2 subunit, or really the region that's responsible for membrane fusion. This is the region that contains that 2p mutation, so preventing the refolding from the pre to post fusion. But if you look on top where the receptor binding domains are engaging with the receptor, you can see that among sarbecoviruses, which includes SARS coronavirus 1, SARS coronavirus 2, and many of the related animal coronaviruses, you can see that there's quite a lot of variation in this receptor binding domain. And so one question we had was, are receptor binding domain antibodies that are neutralizing for SARS coronavirus 2, is there any conservation or is there any ability for cross-reactivity? And so one way that we looked at this, so at the beginning of the pandemic, we of course were trying to find anything that we could that could potentially help therapeutically against SARS coronavirus 2. And the first thing that we did was we said, well, we have mice that have previously been been immunized with the SARS coronavirus 1 spike protein. And we had previously shown, when you have just the SARS coronavirus virus, you get 100% entry. And when you add the plasma from any of the mice that have been immunized specifically with the SARS coronavirus spike, you get 100% blockage of virus entry. And so what that means is that these mice have raised neutralizing antibodies against the SARS coronavirus spike. And so we were wondering, these exact same plasma from these mice that have been immunized with SARS coronavirus 1, how well were they able to neutralize SARS coronavirus 2? And so that's the next experiment that we did. And while you can see it's not 100%, we still have an extremely large drop from entry of SARS coronavirus pseudovirus just on its own, and then addition of this sera, of this plasma, really blocks about 90% of the entry. And so since we knew that majority of the neutralizing capability comes from the receptor binding domain, we were excited to start looking and see what cross-reactive receptor binding antibodies are there, and if there are some cross-reactivity, can we use this therapeutically, and what can we learn about this structurally, and how are they able to neutralize potentially both SARS-1 and SARS-2? And so again, teaming up with Vir Bio, we started looking at human antibodies. So we were looking in the memory B cells to really identify monoclonals that potentially had the ability to bind to both the SARS coronavirus 1 spike and the SARS coronavirus 2 spike. And so that's exactly what you're looking at here. You're just looking at binding, so not neutralization. But what we can clearly see is that there are a number of receptor binding domain directed antibodies that can bind both SARS-1 and SARS-2. And you can see that there's differences between them. So if we just look at this light blue triangle, we have really weak binding against SARS coronavirus 1. But if you look at the light blue triangle against SARS coronavirus 2, it's actually better at binding SARS coronavirus 2. And we have examples going both directions, right? We have this open dark square, which really doesn't bind SARS coronavirus 2, or it does extremely weakly. And yet it's one of the best binders against SARS coronavirus 1. So we're already teasing out clear differences in kind of the landscape of both of these spike proteins. And so we wanted to start picking some of these antibodies to structurally characterize and to figure out, which of these antibodies could really contribute to neutralization. How is it doing that at the structural level? And if these are potent and broad, can we translate them to the clinic? And so the first type of antibodies that we looked at were did we have any that were blocking the receptor binding interaction? So right here in this schematic, I just have the receptor binding domain shown and where ACE2 is binding. And so the first way that we look at this is through biolayer interferometry. And so this is basically just a competition assay. We load ACE2 onto a BLI biolayer interferometry tip, and then we say, okay, then can we dip this tip that's bound with ACE2 into the receptor binding domain? Yes, we get tons of binding because ACE2 is the receptor. What if we preload this receptor binding domain with an antibody that we suspect blocks ACE2 binding? And what you can see in purple here is that if we've preloaded this receptor binding domain with this antibody S2H13, you can no longer see any binding of the receptor binding domain, suggesting that this antibody is really blocking that interface. And we then went on to characterize that structurally. And we found we've now found many, but at this point we found two antibodies that you can see really bind in two completely different ways, but both still block the ACE2 interaction, which is really occurring right at this tip here. And just to go into a little bit of the structural characterization details, so we were able to really look and do some focus classification and symmetry and refinement to really look at the antibody RBD interface, which is really the most important for picking out, how is this antibody interacting with the receptor binding domain. And through this, we were able to really improve the structure and really be able to build in this map. And now we typically find that when the receptor binding domains are closed, we have a much more tractable structural system. But I just want to show that we don't only see the antibodies binding when all the receptor binding domains are closed. In fact, these antibodies require the receptor binding domains to be more open and in a more open position. And so we can also show that when we have a receptor binding domain open, we can see that there's a lot more structural variability, and that it's a lot more difficult for us to really capture this upon cryo-EM, but we can, with extensive classification, get to a moderate resolution of specifically these open receptor binding domains. And so this is just one class of antibody that we can structurally characterize using cryo-electron microscopy. And so we also wanted to see, are there other antibodies binding into different zones that are also neutralizing on the receptor binding domain? And so again, we found kind of another class of antibodies. And so what I'm going to show first is just that we have these three antibodies, one in pink, one in yellow, and one in purple, that are all binding kind of to this outer side of the receptor binding domain, away from, specifically from the receptor binding domain site. However, what we notice is that of these three antibodies, so purple being the farthest from the ACE2 receptor binding domain site, ACE2 receptor site, and pink being the closest, we found that as you got further and further from kind of the interaction between ACE2 and the SARS2 receptor, you got worse and worse at neutralizing the virus, suggesting that potentially one of the mechanisms that these antibodies use to neutralize the virus is really by sterically blocking ACE2 from interacting. and we hypothesize that because as you get further and further away, so this purple really has no interaction with ACE2, it becomes a much weaker neutralizer. Whereas the previous class of antibodies that I showed directly blocks ACE2, that is likely the major mechanism for how these antibodies work. And one other really interesting observation about these antibodies is because these antibodies are really only able to bind when the receptor binding domain is open, something that we've noticed is that, as the receptor binding domains open, they become, the spike protein becomes less stable. And we can actually capture this using cryo-electron microscopy. And what that kind of looks like is we get the release of the S1 subunit. So imagine chopping the protein kind of right in the middle here. And what you can see, and this is just one example of one of these classes of antibodies, you can see in light pink and dark purple that the antibody is still bound in this S1 released structure. And we kind of see this with all of these antibodies that require the opening of the receptor binding domain. And so you can see the trimer of the spike protein in yellow, blue, and light pink, and then you can see where the antibody has really engaged on the top. And what's interesting to us is this is happening, even though we have a 2-proline stabilized spike protein, really suggesting that these antibodies are driving and forcing the receptor binding domains to stay open and in a really unstable position, which is potentially another mechanism or way that they kind of, quote, destroy the spike protein. There's one more class of antibody that we really have identified through all of this work with VIR, and that is one that binds to kind of this front side of the receptor binding domain. And again, it's very far away from the ACE2 receptor binding site. And so what's really interesting about this antibody, and this antibody is called S309, is that it neutralizes extremely potently without blocking the ACE2 receptor interaction. And it also binds to an extremely conserved site on the receptor binding domain. And this antibody has done really, really well in clinical trials, and what's really exciting is that in this zone, there's no major mutations that have been popping up naturally among circulating SARS coronavirus 2 variants. And so through all of this structural characterization, so we've kind of identified these 3 classes of antibodies, we've, through cryo-EM, made an antigenic map and basically defined a landscape of neutralization of SARS-2 with these varying classes of antibodies. And so with all of this knowledge, we kind of started to ask ourselves, can we make a vaccine that takes the information from these antibodies that occur naturally in patients who have been infected either with SARS-1 or SARS-2, and can we take this information and really build a vaccine that targets these known sites responsible for 90% of neutralizing activity and really highlight that in our vaccine? And so that was kind of the question that we set out to address, and we teamed up with Neil King and the University of Washington Institute for Protein Design really to do this. And again, I'm going to show a video from Janet Owasa, just kind of displaying what we've done here. And so again, we have the SARS coronavirus 2 virus kind of displayed here with the spike proteins on the surface, glycosylated, and with their receptor binding domains moving from an up and a down presentation. And so we knew that we wanted to highlight the spike in our vaccine, and specifically the receptor binding domain. And so what we did is we fused this receptor binding domain to a scaffold, which trimerized the receptor binding domain. And then in collaboration with Neil King's group, we put this scaffold onto an existing nanocage. And so this nanocage is made with 2 designed components, one in gray, which is the trimeric component, and one in orange, which is a pentameric component. And so what this allows us to do is really to display 60 copies of the receptor binding domain on a nanocage platform to use as an immunogen. And I'm just going to show that kind of a little bit slower here. So we have 20 copies of the receptor binding domain of SARS-2 fused to a trimer, and then we have the pentamer that assembles into this beautiful nanocage. And we can look at this by negative stain electron microscopy, and we can characterize it also by cryo-electron microscopy and see really that we have these beautiful homogeneous nanocages that are displaying the receptor binding domain. And what we showed towards the end of last year was that this receptor binding domain elicits extremely high and potent neutralizing response in mice. And so what I'm showing here is the receptor binding domain on its own, all the way to the left, is not able to elicit a neutralizing response, at least at these low doses. And so the goal of using low doses is you have more vaccines for more people. The spike on its own, so the S2P trimer, SARS-2 spike, is able to elicit a neutralizing response, but only at a higher dose, so at 5 micrograms. And no matter what we did to the nanocage, whether we had a short or a long linker, and whether we had a low valency, so 50% of the receptor binding domains occupied, or 100, we really were able to get high neutralizing titers against SARS coronavirus 2 at extremely low doses, so up to 1 microgram of the nanocage. And this was doing much better than just the S2P spike on its own, and much better than native infection, which you can see here in orange with the human convalescent sera. And not only were they able to elicit highly potent neutralizing titers, they also protected mice from infection with mouse-adapted SARS coronavirus 2. So what you can see is that the receptor binding domain has full infection. There we can detect viral RNA copies in the nose and lungs. The low dose of the SARS-2 spike, the 2P spike protein, has some breakthrough as well. The high dose of the spike protein is fully protected, and all doses and all iterations of the receptor binding domain nanoparticle are fully protected in mice. Now, not only are they fully protected and eliciting really high titers at a low dose, but we also showed that we're really targeting many different locations on the receptor binding domain nanoparticle. So we're getting antibodies that block ACE2, which we can see through this BLI competition assay, where as we add purified polyclonal Fabs, we can actually block the binding of ACE2, which the native binding is in black here. We can do the same thing with CR3022, which is mimicking similar style of antibodies to the 3 that I showed earlier. And we can also show this for S309, which is the back end receptor binding domain antibody that is moving quickly through clinical trials. And so we can elicit this really broad response. And the goal of knowing that we have this broad response is, we have all of these variants coming, kind of popping up everywhere. And so the kind of goal of this type of vaccine is, if we have a broad response, especially we're eliciting antibodies like S309, which bind to a zone that has so far not been mutated, the goal is that no matter what antibodies no matter what variants are coming, that we have antibodies that are broad enough to still prevent infection even in the face of these mutations. Now, that's not the only way that we're trying to make this vaccine broad and ready for potential variants. We're also making what we call mosaic or cocktail receptor binding domain nanoparticles. And so the way that we're doing this is we're putting various receptor binding domains. So we have SARS-2, which was our previous vaccine, which is currently in phase 1 clinical trials. But we're also putting other related sarbecovirus, or family of SARS-1 and SARS-2 receptor binding domains on this nanoparticle platform. And so we've chosen in this first iteration 4 different receptor binding domains. So SARS coronavirus 2 receptor binding domain, SARS-1, and then a SARS-1 related bat coronavirus, WIV1, and then a SARS-2 related bat coronavirus, RATG13. And so we've put this into both a mosaic, which means that we have a mixture of all of these receptor binding domains present, on a single nanoparticle. And we've also tried to see in cocktail form, we have just RATG13, just SARS-2, just SARS-1, and just WIV1 all injected into mice at the same time, and really to kind of identify how this is going. And so I just want to end with one final video showing that we're currently testing these mosaic and cocktail nanoparticles, but the goal is really to see, like, we have all of these variants coming out, and if we can put a variety of these receptor binding domain variants onto a single nanoparticle, so this is showing an example of a mosaic nanoparticle, the goal would be that we elicit broad neutralizing antibodies, not just against whatever sequence is present in the vaccine, but also antibodies that can tackle the next variant that we don't even know whether it's come out yet. So the goal is you have these broadly binding antibodies that can really block entry of the virus, even if the vaccine doesn't even contain these receptor binding domain strains. And so we just put this on bioRxiv, and we're excited to see where this goes. But this is kind of the future of how we use structure to understand antibodies to then take these structures and take these interactions of known neutralizing antibodies with the receptor binding domain and push them into a clinical vaccine candidate. And so just to briefly summarize what I've talked to you about today, kind of what we've done over the past year is we've mapped monoclonal antibodies using cryo-electron microscopy and X-ray crystallography to the receptor binding domain, the region where 90% of neutralizing activity against SARS coronavirus 2 is. And we've used this to propel monoclonal antibodies into the clinic and also a vaccine candidate into the clinic. And this would not have been possible without first determining the structure of the SARS coronavirus 2 spike in its multiple conformations and understanding that there's really a wealth of data showing that there are cross-neutralizing antibodies that target the receptor binding domain, and these are potent, and we can bring these towards therapeutic uses. And so with that, I first have to thank everyone in the VISA lab. This honestly is even an old photo. We've continuously been growing over the pandemic, but I really want to highlight. So Young has done a lot of the structural characterization work that I've shown today, and then of course David has been a fantastic mentor. We've worked together for the past 5 or 6 years, and he's really taught me everything I know about cryo-electron microscopy and about scientific discovery. And then I've also shown you a lot of work that has been collaborative, with a lot of different groups. And so I just want to highlight the major players here. So I talked a lot about our work in collaboration with HuMabs and Vir Bio, where they do a lot of antibody discovery and characterization, and we've been doing a lot of the structural characterization and biophysical characterization. We've worked with collaborators at Fred Hutch here in Seattle, and then of course our large collaboration with the University of Washington Institute for Protein Design to really get these protein designed nanoparticles displaying the receptor binding domain into phase 1 clinical trials with their design. And then the challenge work that I showed was done in Ralph Barrett's lab at the University of North Carolina. And I will end there and just remind everyone how to submit questions, I think. And I'm happy to take any questions that you all may have.
Yeah, thank you, Lexi, for such a beautiful talk. Again, I would remind everyone present here today to submit your questions by typing in the question panel right here, as Lexi was suggesting. And again, do not hesitate to ask any questions, anything about latitude or anything that I presented or anything Lexi presented. So Lexi, we do have some questions here, if you don't mind answering. So the question is, how do the variants or mutations in SARS-CoV-2 virus would affect the landscape of the monoclonal antibodies or the vaccines that has been developed so far?
Yeah, so this is something that pretty much everyone in the field is thinking of, and honestly trying also to predict in a way. The virus, up until kind of mid 2020 seemed to have kind of a stable sequence, and since then we've kind of, with increased sequencing efforts and a lot of work done, we've been finding that there are a lot of novel variants really coming out. And so there are a lot of variants that we're all kind of noticing and worried about. So the first is B117, which originated or was originally sequenced in the United Kingdom. And this sequence has kind of been taking over because it has been shown to have slightly increased infectivity. Now, the increased infectivity and potentially also increased clinical signs is really detrimental. However, the mutations in the spike protein really are not affecting the landscape of a immune response. They do knock out some of the monoclonal antibodies. So there's a mutation on the receptor binding domain called 501Y. Some of the monoclonals are knocked out by this, but overall, an immune response from a vaccine or from prior infection seems to still be able to handle this B117 totally fine. That is not true for all of the variants, though. There are a couple of variants, so one named B1351 and one named P1. So these originated or were originally sequenced in South Africa and Brazil, respectively. These have a kind of a smattering of mutations that does seem to lower the overall immune response. Many of the monoclonal receptor binding domain antibodies are knocked out through these mutations, and there does seem to be a drop in immune response. So these are kind of the ones that we're looking at and we're worried about. And then there's even more coming out like daily that we're really trying to focus on. So another one that we've characterized is B1427, which was originally sequenced in California. Again, certain of the monoclonal antibodies are completely abrogated, not just in the receptor binding domain, but also in the N-terminal domain, which I didn't have time to talk about today, but also has neutralization capability. And again, kind of the polyclonal response, so after vaccination or infection, there is a drop from this B1427 that was originally found in California. So we're constantly learning, but that's kind of why we in the field are looking at monoclonal antibodies like S309, which bind to a region that so far we haven't found naturally occurring mutations to, or we're really driving this, this receptor binding domain nanoparticle. We're showing that we can have broad responses, and we're also trying to even broaden those responses further for future vaccines. So we're aware of it and we're working on it.
Well, thank you very much for answering this question. Let's see if we have another question. What challenges did you face with respect to SARS-CoV-2 spike protein with and without the antibody fragments or FABs in terms of sample heterogeneity?
Yeah, so kind of, I'll just go back to this slide. So kind of the first aspect of sample heterogeneity that everyone in the coronavirus field is dealing with is, the core of the spike will refine to extremely high resolution. However, the N-terminal domains and the receptor binding domain, so receptor binding domain, I have this up arrow to, and then directly next to it is the N-terminal domains. And these are really flexible. The receptor binding domain is literally moving from a down to an up conformation. And as a result, the whole S1 subunit is really breathing, making it a really challenging structural target. Now, we can use focus classification and honestly extensive classification to really put all of the particles that are fully down into one bin, use a C3 symmetry to refine. But the different receptor binding domain characterizations, these have to be done with C1 symmetry, and the refinement is definitely not as high of resolution because of that. In some instances, having an antibody fragment helps stabilize this segment, and in some instances, it gives it even more motion. So it's kind of a mixed bag whether they help or hurt certain zones, but definitely extensive classification and focused classification is kind of our friend here.
All right, thank you, Lexi. We have another question. How will your vaccine be delivered? For example, with respect to other vaccine from mRNA, Pfizer, or Johnson & Johnson?
Yeah, so right now it's kind of the exact same method, so intramuscular. We would love to have, a fully aerosolized spray and working towards it, but definitely for right now, similar injection to the mRNA or adenovirus platforms. And we're currently testing 2 doses, so basically exactly the same as all other vaccines on the market right now.
Okay, I think there's a question about Latitude, so I'm going to be answering that. Is Latitude compatible with Joule microscopes? Yes, Latitude S and T are compatible with Joule cryo-arm systems, 200 and 300 kV, and they're compatible, and Latitude is compatible with almost all FEI microscopes as well. So Lexi, I have another question for you.
Yeah.
Have you ever compared your RBD nanoparticles versus the currently available vaccines?
Yes. So we have looked just at human sera from vaccinated individuals like Pfizer and Moderna. It's hard to directly compare since none of our samples are currently we do not have access to the human samples yet. But what we have been doing is benchmarking against similar style vaccines. So we've been benchmarking against protein SARS-2 spike or protein hexapro, which is an even further stabilized spike protein. And what we see is that the immune response, at least in non-human primates, is very, very similar. So we're getting to similar neutralization titers, similar amounts of breadth. So, against different variants, the receptor binding domain looks very, very similar to a spike-based vaccine. And we think the reason for that is, one, that most of the neutralization capacity is coming from the receptor binding domain, and 2, because we're presenting just the receptor binding domain, we may even be getting more benefit of types of antibody, like breadth of receptor binding domain style antibodies, and that may be an advantage over some of the spike protein based vaccines. But yeah, it's a great question.
Thank you, Lexi. So for the sake of time, I think we'll take the rest of the questions offline. Again, if you have submitted any question, we'll be sending you an email with the answer to your question. With that, I thank you, Lexi, for giving such a beautiful talk, and I thank everyone present here today for their time and attending the webinar series. Thank you.
Thank you so much.